the faculty, we are extremely proud that you are here, that you are participating in this 25th Krakow Conference on Endothelium, initiated by Professor Ryszard Gryglewski, a famous researcher from our Jagiellonian University. I looked into the program. I know that this is a fascinating program with extremely interesting lectures and abstract presentations, mostly on basic research on endothelium, and that's why you are here, through the translational research to improve the prognosis of the patients with vascular disease.
So 25th Krakow Conference of Endothelium is open. It was started really in '93 and every year about endothelium, and still we think that's something to talk about.
The first meeting really was the dawn of what we have begun to do with the endothelium. That was a meeting that was centered on nitric oxide. And now, over 25 years later, we have identified multiple roles that the endothelium plays. We have found many cells that interact with the endothelium that we didn't know about. We have identified dozens of mediators that change the way the blood vessels react. All of this has happened over the 25 years that these meetings have gone on. And most of it, much of it, has been presented here. When you realize that how many Nobel Prizes have been involved in this, that's how important the endothelium is. I think it's well recognized that the endothelium is an organ. So I think we're at a point now where the endothelium, instead of just being a lining that kept things from leaking out of a blood vessel, is now an incredibly important part of how organs work and how we control blood pressure and how we regulate the way the body works. The endothelium is very, very important.
The problem with it is that if these cells are different, that means they express different signaling pathways, different proteins, so when you hit them with a drug, they might respond differently. And it's hard to test the drug on every single vascular bed. There's also a chance, because it means that basically almost every vascular bed has a different array of surface proteins. And there's some really beautiful papers showing that basically you can pin a particular vascular bed with a zip code of proteins on its surface. So if you want to target that with a drug and make sure the drug doesn't stick anywhere else, then you have a chance of delivering drugs.
According to large population epidemiological studies, there is clear evidence, meanwhile, that noise exposure increases the risk for cardiovascular disease, cardiovascular events, and in the worst case also cardiovascular mortality. Clean noise exposure affects the function of the endothelium in a way that you create oxidative stress, that you have increased inflammation by infiltration of immune cells to the vascular wall, and thereby definitely you have impaired endothelial function. So definitely in the future doctors also have to take care on these risk factors and not only on the traditional ones like hypertension, diabetes or obesity.
There are many studies indicating that there is not only an impact of the microbiome on vascular or endothelial or metabolic function, but also vice versa. So disease states that change the metabolics, that change blood pressure, like diabetes or obesity, also impact on the composition of the gut microbiome. So it will be a first task to really disentangle this mutual relation between the microbiome and the body to really find the proper targets to then develop new strategies to maybe treat this. So there will be a long way to use these findings also for human medicine.
By research on circadian clocks, we can understand the different of physiology between day and night, and this can be very important for endothelial function, which is known actually to differentiate between day and night. So I believe that by studying the circadian clock, we can identify some novel functions of the endothelium which are underappreciated when we study them only during the day and not throughout the 24-hour cycle.
I think NOx4 is special because it really emphasizes this concept that reactive oxygen species can mediate important, protective, beneficial signals. So we have known this, people have known this for a long time, but now we have a very specific molecule, enzyme, which is involved in some of these protective pathways. So I think it's important, one, because it exemplifies the concept, and secondly, of course, if we can define how NOx4 works, we may be able to identify some therapies, some therapeutic interventions that are more sophisticated, where we can enhance the protective pathways instead of just trying to inhibit reactive oxygen species. That's the potential.
The biggest challenge of not just endothelial biology, but cell biology, is that we are making a lot of beautiful little puzzle pieces and not enough effort in putting the puzzle together. We don't study enough processes coupled together as much as we need.
We should perhaps stop looking for one thing or one tissue or one organ that's more important than the others. We know this in terms of signaling, but maybe the same is true in terms of the endothelium. You know, we need to think about how it works in partnership with other cells and tissues. So if you like, we should think, I personally believe we should think more in terms of systems biology rather than just looking at one organ or one tissue or one signaling pathway.
I believe that these small conferences like this one, where researchers from all over the world are invited and really convene in a small setting, is the perfect way of scientific exchange.
Professor Gryglewski always brings together a lot of very interesting and exciting scientists, and already this morning I learned a lot from the other colleagues giving the talks.
I'm so excited and impressed by the number of students who are here. It's a remarkable opportunity for young people, and it's a great opportunity for established investigators to get new ideas.
What I found very special in this meeting today, actually, was the last session we just had with the 1-minute presentations by younger, earlier career scientists. I thought this was wonderful.